Hello everyone, good morning. So we are going to continue in the notes from my class that the professor went over for the test. And this time we're going to go into the second half of adaptive immunity, where we're going to look at the cell-mediated immunity or the T cell response. So this is going to be different from the humoral response or the humoral system that we went over last time. This time we're not producing any antibodies in cell-mediated immunity. And we're going to be looking at the T cells more than the B cells. That being said, so the T cell, again, does not make antibodies. And the effector T cells are actually going to interact with the target cells to cause a change. Alright, next, we're going to talk about how they mature in the thymus. There are two subsets to help eliminate antigens. They have different surface proteins. You first have the cytotoxic T cells, or TC, and you also have helper cells, TH. These are going to have multiple copies of the T cell receptor, or TCR. And what's interesting about the TCR we were discussing earlier in the humoral system, where we're going to have this presentation done when we're doing a B cell activation with a T-dependent antigen. We're going to go and get the fragments that are degraded, and then the fragments are going to be displayed on this cell by the MHC2. So that's the histocompatibility complex. And then those are going to be recognized by the TH cell. They're going to be recognized by the helper cells, TCR, the receptor. All right. And then a third subset also exists, the T regulatory. This one, they do have TCR, they do have those receptors, but the role is to prevent immune system from mounting a response against the self. Alright, let's talk about the activation of the helper and the the cytotoxic and then the helper cells. So these need to be activated before they can multiply. They're going to be activated by the dendritic cells. Then once they activate, that's going to cause proliferation and differentiation. The effector TC, these are the effector cytotoxic T cells. They're going to respond to intracellular antigens, especially those that tell the cell to go into apoptosis. Whereas the effector TH cells will help the humoral and the cell-mediated immunity. When it's helping the humoral, this is going to activate, the TH effectors are going to activate the macrophages, B cells. They're going to also make the cytokines. And they're also going to support other T cells. Both types. Both of these could actually form memory cells as well. Alright, so how does this work? We're going to have our naive B cell again on one side. That's going to be kind of our innate immunity. However, then we split off into the adaptive immunity, the humoral system, and then also the cell-mediated. So there's a little bit of a chart here. So innate immunity will be the dendritic cell. And then the dendritic cell will activate the T cells. Let me move this over so we have more room. We're going to activate the T cells that bind antigens representing danger. So we're going to have a naive helper as well as a naive cytotoxic T cell. So this is going to have them proliferate and differentiate once they're activated by the antigens representing danger on the dendritic cell. Remember, we need to present it to the T cell in order for it to continue to function. So that's going to differentiate and proliferate both the cytotoxic as well as the helper cells. Then we get to cytokines. Again, the voices of the cell, and that's going to affect a naive B cell. Once the cytokines are recognized by the B cell, there's going to be proliferation and differentiation of that B cell as well to make plasma. And then again, only the B cell that has the specific receptor for the specific antigen at that time. That's the only one that's going to proliferate and differentiate. And then once it gets to plasma on the B cell, again, this is the humoral system. We're just doing a little bit of review, connecting everything. We're going to produce the antibodies. So that's how the CMI and the humoral system are connected. Now on the right side, when we had those TH cells, we're also going to, again, get some of those cytokines to affect some macrophages. And that's going to increase their power. With the TC cells, however, they're going to deliver death packages. And when they deliver a death package to a self cell that is infected, this is going to cause death to that self cell. So apoptosis. Okay, so let's again talk more about the T cell response. Again, the T cell receptor does not bind with free antigen. We talked about it earlier where it's kind of just half of that antibody. Looks like that. And then there's something called the CD. There's the membrane. So the TCR, it can't bind with free antigen. And it only acts when there's presentation by another cell. We are going to be using the major histocompatibility complex, the MHC. And there are two types. We talked about class 2 already. So class 2 of the MHC, this is going to present exogenous antigens. Exogenous means they were taken up by the cell. And these are produced by antigen presenting cells, which include the dendritic, as well as B cells and macrophages. It's number 2. And then exogenous. Exogenous is number 2. Remember that. Right? Type 1 is something different. These are going to be endogenous antigens. Endogenous means that it's made within the cell. These are going to be found in all nucleated cells. Made in the cell is type 1, whereas type 2 is the exogenous antigens. It's taken up by a dendrite, a B cell, or macrophage. And again, presentation, that's how we get the response. Okay, so how do we activate them? So first we could use a dendritic cell. Again, either MHC1 or MHC2. MHC2 is going to probably take preference here because, again, it's exogenous material. Dendritic cells are going to be in the peripheral tissue, such as the skin and mucosa. They're going to gather by phagocytosis, and then sample material between epithelial cells and mucosal barriers and in respiratory tract, and the lumen of the intestines, a sample of epithelial, of mucosa, as well as respiratory and the intestine, the lumen specifically. We also have toll-like receptors, or TLRs. That's another abbreviation. Again, toll-like receptors are actually seen in the innate immunity. If the pathogens are found, and then that's going to increase intake of material. They go to the secondary lymphoid organs, such as the spleen and stuff. They're going to make the co-stimulatory molecules. And these mean danger. And then this is going to present antigens. The TLRs are going to present the antigens by both MHC1 and 2. So endogenous and exogenous antigens, and then this is going to activate both TC and TH. Macrophages and B cells are also going to activate them. But they only present by MHC class 2, as we discussed earlier. And then they could also make those co-stimulatory molecules as well. Okay, so this image here in this slide, it's just showing a dendritic cell again. And one of the scenarios is exogenous, one of them is endogenous. So endogenous would be like that particle there that's from the cell itself, and then we have the TC and the TH. Whereas maybe it took in something or phagocytosed a bacteria, so now it's displaying that material. That's going to be that's going to be the class 2. So this one's class 2, this one's class 1. Class 1, again, it came from the dendritic cell itself. And then class 2, that's going to be from an outside source. The TC cell is also known as the Cd8 cell, while the CD4 is going to be the TH, the helper. Right, now we're going to look at the TC, or the CD8 cell, in detail. So the cells are going to present internal antigens, or proteins, endogenous antigens in MHC class 1. And this binding TC will indicate the recognition of pathogen or cancer. Then this is where we're going to get apoptosis. And this is going to just secrete a ton of enzymes, granzymes, proteases, cytotoxins. Perforin is one of them. Right, and then the TH, or the CD4 cell, is a bit different. This one's going to find exogenous this time. Then, of course, that's tied to MHC class 2. And then these are going to come from antigen presenting cells, which of course are dendritics, macrophages, and B cells, other phagocytic cells. The naive B cell binds to the antigen, and then the TH, or the CD4, will deliver cytokines. When it finds the MHC class 2, and then the cytokines activate the B cell. And then again, B cell, they'll become only the specific one that has a specific receptor to the specific antigen at the time. Then it becomes plasma, and then we have the effectors, which are plasma, and then we also make memory cells, and then those antibodies. A hapten, this is a molecule too small to cause a response unless it's connected to a protein. Example would be penicillin causing allergic reaction. Right, so some things highlight here. TC as well, apoptosis is the TC, while TH is activating the naive B cell. Okay. Again, the macrophages will engulf and degrade the foreign material, then present on MHC class 2 molecules on their surface, a macrophage, a B cell, or dendritic cell. T cells will recognize these peptides, these protein fragments, on the MHC class 2 and release cytokines to activate macrophages with increased power. We also have this third category of the lymphocytes, the natural killer cells, which we haven't talked about much. And these are going to cause apoptosis in the antibody. Let me write that better. Antibody bound self cells. And these are going to find hosts that are containing these foreign proteins in the membrane bound by antibodies. The antibody-dependent cellular cytotoxicity, this is what it's called, the ADCC, antibody-dependent cellular cytotoxicity. The NK cells have the Fc receptors for IgG molecules. Again, if we go back to our chart when we were going over the humoral system, the IgG are going to be most seen in tissues, and they're going to activate the complement system. Again, it's just another type of antibody. There's, again, Migande, so MGADE. We're just talking about IgG. NKs also bind to the antibodies, and then they deliver perforin and protease granules to the cell. These are, again, those enzyme packages, basically. And then, of course, this causes apoptosis. They also recognize host cells lacking MHC class 1. MHC1, again, those were the endogenous antigens that are going to be recognized by those. But the natural killer cells will also find host cells lacking MHC class 1. Okay, so in review, I have this picture from when I was taking the notes in class. So if we have an immature T cell, once that's activated by a dendritic cell, we're going to, of course, get the TC and the TH. The TC are going to be active by the dendritic cells again, and they're going to mark host cell with a virus infection and bring the death package. The TC cells are. The natural killer cells are also going to just get the infected self cells, but then that process is a little bit different. We just went over it. The TH cells, the T helper cells, or CD4s, are going to find the antigen on the MHC of the B cell, and then this is going to cause the memory helper T cells, and then they're going to activate the macrophages when they recognize the macrophages antigens. Again, it's displayed on the surface. We need presentation. And then if we go to the naive B cells, this again is going to be part of the innate immunity a bit. We're going to touch on that, but then we also have the B cell activation. So that's going to be humoral system. So the naive B cell plus the antigen, the specific receptor, and then plus the antigen again. It's naive since it hasn't found that antigen yet, but once it does, this is going to cause the differentiation and proliferation into the plasma effector cells as well as the memory B cells, and then you get your antibodies. Alright, thank you so much for tuning in. That was basically everything from the adaptive immune system on the CMI side of things. Take care and have a good one. Please do something nice for someone.